lahan kering dan basah yang ramah lingkungan dan berkelanjutan, misalnya pengolahan limbah dan hidroponik. Yang ketiga, rekayasa bioproses dan yang menangani pengolahan hasil-hasil pertanian, misalnya pemanfaatan teknologi near infrared dan teknologi spektroskopi, contohnya untuk pengolahan kopi. Kemudian juga penanganan nanoteknologi di bidang pertanian dan pangan. Yang keempat, sistem dan manajemen keteknikan pertanian bisa meliputi antara lain teknik dan ekonomi penanganan. Yang ketiga, penerapan rekayasa bioproses, pengembangan usaha menangani jasa hasil pertanian, serta sistem dan manajemen pengelolaan hasil teknologi spektroskopi.
Jurusan Teknik Pertanian memiliki 3 bidang minat dan 3 laboratorium. Yang pertama adalah laboratorium daya dan alat mesin pertanian. Yang kedua adalah laboratorium rekayasa sumber daya air dan lahan, dan yang ketiga adalah
penerapan rekayasa bioproses, pengembangan usaha menangani jasa hasil pertanian, serta sistem dan manajemen pengelolaan hasil teknologi spektroskopi.
Jurusan Teknik Pertanian memiliki 3 bidang minat dan 3 laboratorium. Yang pertama adalah laboratorium daya dan alat mesin pertanian. Yang kedua adalah laboratorium rekayasa sumber daya air dan lahan, dan yang ketiga adalah
penerapan rekayasa bioproses, pengembangan usaha menangani jasa hasil pertanian, serta sistem dan manajemen pengelolaan hasil teknologi spektroskopi.
Jurusan Teknik Pertanian memiliki 3 bidang minat dan 3 laboratorium. Yang pertama adalah laboratorium daya dan alat mesin pertanian. Yang kedua adalah laboratorium rekayasa sumber daya air dan lahan, dan yang ketiga adalah
penerapan rekayasa bioproses, pengembangan usaha menangani jasa hasil pertanian, serta sistem dan manajemen pengelolaan hasil teknologi spektroskopi.
Jurusan Teknik Pertanian memiliki 3 bidang minat dan 3 laboratorium. Yang pertama adalah laboratorium daya dan alat mesin pertanian. Yang kedua adalah laboratorium rekayasa sumber daya air dan lahan.
Usaha menangani jasa hasil pertanian, serta sistem dan manajemen pengelolaan hasil teknologi spektroskopi.
Jurusan Teknik Pertanian memiliki 3 bidang minat dan 3 laboratorium. Yang pertama adalah laboratorium daya dan alat mesin pertanian. Yang kedua adalah
penelitian. Banyak penelitian yang dilakukan di sini, seperti analisis mesin, pembuatan pupuk pelet, pembuatan papan partikel, dan juga pengeringan bahan-bahan hasil pertanian seperti vanili atau lada yang sering dilakukan di sini.
Baik, sekarang kita sudah di laboratorium yang kedua, laboratorium B, yaitu laboratorium rekayasa sumber daya air dan lahan. Yuk kita masuk. Halo Kak.
Hai, selamat datang di laboratorium LB, laboratorium rekayasa sumber daya air dan lahan. Di sini ada beberapa alat yang sering digunakan pada praktikum di LB ini. Ada yang pertama itu ada magnetic stirrer, di mana magnetic stirrer ini berguna untuk mencampur atau mengaduk bahan pada tabung Erlenmeyer. Yang kedua itu ada prelabo. Prelabo ini berfungsi untuk memanaskan bahan untuk mengambil atau penyulingan. Yang kemudian di belakang saya ada lemari penyimpanan, di mana lemari penyimpanan ini untuk menyimpan alat-alat laboratorium yang tidak begitu digunakan. Kemudian di sebelah kanan saya itu ada tanur, di mana tanur ini berfungsi untuk menguji kadar abu pada suatu praktikum atau suatu penelitian. Yang kemudian ada oven yang berguna untuk menentukan kadar air pada suatu bahan. Kemudian di situ ada timbangan, berguna untuk menimbang suatu bahan atau suatu alat yang digunakan pada praktikum. Terima kasih.
Baik, sekarang kita sudah di depan laboratorium yang ketiga, yaitu laboratorium C, tentang laboratorium rekayasa bioproses dan pasca panen. Mari kita masuk. Halo Kak.
Hai. Ini adalah Laboratorium Bioproses dan Pasca Panen atau disebut dengan Lab C. Dengan kegiatan yang biasanya dilakukan di lab ini yaitu seperti kegiatan teknik penyimpanan, teknik pengeringan, teknik pendinginan, dan teknologi pengolahan hasil produk pertanian dengan didukung dengan alat-alat seperti oven, UV spektroskopi, penetrometer, dan sebagainya.
Jurusan Teknik Pertanian Fakultas Pertanian Universitas Lampung mempunyai sebuah organisasi kemahasiswaan bernama Persatuan Mahasiswa Teknik Pertanian atau yang biasa disebut dengan Permatep. Yuk kita cari tahu lebih lanjut tentang Permatep. Halo Kak.
Persatuan Mahasiswa Teknik Pertanian atau Permatep merupakan organisasi kemahasiswaan di jurusan Teknik Pertanian. Permatep memiliki berbagai macam program yang menunjang kegiatan-kegiatan kemahasiswaan di jurusan Teknik Pertanian yang selalu bersinergi dengan jurusan Teknik Pertanian. Beberapa program kerja yang diemban oleh Permatep yaitu ada Desa Binaan dan Minat Bakat. Banyak sekali prestasi yang sudah diraih oleh rekan-rekan kita mahasiswa jurusan Teknik Pertanian, yaitu ada juara 3 Lomba Inovasi Teknologi Pekan Teknik Pertanian Nasional yang diadakan di Politeknik Negeri Payakumbuh, Sumatera Barat pada tahun 2019, dan rekan-rekan mahasiswa juga aktif dalam kegiatan program holistik pembinaan dan pemberdayaan desa yang dibiayai oleh Kemendikbud, dan masih banyak lagi prestasi-prestasi yang diraih oleh rekan-rekan mahasiswa jurusan Teknik Pertanian. Inilah kami, keluarga besar jurusan Teknik Pertanian. Mari berproses bersama kami dan berprestasi bersama kami. Tektan Jaya, Permatep bisa.
Assalamualaikum Pak Kajur.
Selamat pagi Mr. Diding. Sehat ya?
Pagi ya, alhamdulillah sehat.
Pak, untuk Pak Kus sudah ya?
Sudah.
Sudah.
Pak Wadek. Masih di mute Wadek. Assalamualaikum.
Assalamualaikum.
Waalaikumsalam.
Assalamualaikum semua.
Salam, sehat Pak Wadek.
Alhamdulillah, sehat Mas.
Pak Wadek kelihatan tambah montok sekarang paginya. Habis sarapan kali ya?
Pak Kajur sudah ini tambah segar Pak Kajur.
Seger, susunya cocok Pak. Buatun,
Jur.
selamat pagi Buatun.
Pak Kajur,
Ya.
Pak Sandi.
nanti saya sudah ngasih sambutan mewakili Pak Dekan ini langsung keluar saya ya.
Oh ya, enggak apa-apa ya.
Karena jam 9:30 ini juga saya ada rapat dengan WN juga bahas
Ya,
pertor kemahasiswaan. Jadi nanti mohon maaf.
ya.
Ya.
Ya enggak apa-apa Pak, orang
Oke, aku udah ngomong sama Pak Warji tadi.
ya sudah sip pokoknya.
Siap Pak.
Jalan terus. Yang penting sambutan sampean pembukaan ini bikin Pak Jano semangat nanti. Kalau nggak ada sambutan Bapak, Pak Jano nggak semangat nanti. Dokter Jano.
Masya Allah. Ini mas seminar kerjanya segini banyak.
Diding kopiannya agak miring dikit kenapa?
Ya, memang sengaja miring. Sengaja Pak miring Pak.
Iya, kurang miring dikit kopiannya. Awas.
Assalamualaikum Bu, sehat Bu ya?
Alhamdulillah sehat Pak. Pak Ketuanta gimana sehat?
Sehat Bu, insya Allah sehat.
Mudah-mudahan kita semua sehat.
Tadi nggak ikut pemberangkatan Pak Anu ya Pak? Pak Sarno.
Ini kali. Itu ya.
Tadi saya nggak ikut Mas,
Iya.
kayaknya dibawa.
Pagi juga, ke pagi juga jam 8 udah berangkat tadi.
Udah, karena habis subuh itu sholat kan berangkat itu.
Iya.
Ya, Pak Suara sadar.
Sampai sana udah berangkat tadi. Waduh ya sudah.
Iya.
Kecil ya.
Bukan kalau kecil nanti.
Ingat
Coba ke
aja.
sini. Ke saya ya. Udah muncul.
Ini nama Pak siapa ini yang memberikan materi ini?
Pak
Dr.
Jano
Stephen aja Pak. Kalau
Stephen.
susah, Dr. Stephen gitu.
Stephen.
Dani Stephen.
Jane Stephen Dehia gitu ya.
Iya.
Iya.
Namanya susah.
Susah Bu. Dani Stephen Iya.
Pakai di lidah kita, Pak.
Iya, Bu. Meliput lidah kita, keliput-liput.
Nah ini undangan banyak benar yang di Bukit Randu itu pada enggak bisa kayaknya Pak.
Iya, ini kan pagi ini kan
Pagi ini.
iya ada yang di Bukit Randu. Kalau Pak Dekan ke BPTP yang beri materi Pak Dekan.
Iya, Pak Dekan.
BPTP yang ngisi materi.
Ini tak banyak teman-teman kaji juga pada nggak bisa semua ya sudah kita di kampus.
Belum. Masih tidur.
Ini kali ini. Ya.
Beli ayam. Iya.
Rotet.
Ayam.
Pak Sabto, selamat pagi Pak Sabto.
Kalau yang lain, sudah yang ini, yang ini coba. Bukan, Pak. Bisa, ada mulai. Saya salah kostum lagi, tak pikir hari ini Kemis. Batik. Mau balik lagi. Terus tanggung ya masih di kampus.
Belum jadi
Waktu makan uduk tadi kekenyangan Mas Ding, jadi lupa.
Bukan uduk tadi. Nasi putih. Iya.
Ini hari Rebo, Mas. Udah 112 ini partisipannya nih.
Makasih ya, Bu.
Dan ini tempatnya ya, tempatnya Mas Diding ini Mas ya? Untuk sekolah dulu ya?
Ya, ini adik kelas saya Pak. Jadi saya itu selesai doktor 2013, dia 2014 masuk master. Masih sangat muda ya. Tapi saya karena hampir tiap tahun ke sana, jadi saya ketemu gitu sampai terakhir 2019 saya ketemu di GIFU, dia sudah jadi peneliti tamu di GIFU.
Oh, di GIFU ya.
Di tempatnya Pak Nakano itu.
Kebangsaannya apa itu?
Ya?
Kebangsaannya apa?
Kenya Pak, Kenya.
Oh, Kenya.
Lahir di Nairobi, Kenya. Kemudian kuliah di Kenya juga, S1-nya, S2, S3 baru di Kyoto. Terus postdoc di GIFU. Nah, terus sekarang kerja di Naro. Stephen Sang. Dr. Stephen.
Good morning, Dr.
Good morning.
Yes, how are you doing?
I'm fine. How are you?
I am doing well. It's good to see
Okay.
all of you gathered for today's lecture. Thank
I'm
you
glad
for inviting me.
to see you again. Yesterday, we had a very nice Zoom connection test. Okay.
Yes. Thank you for that. Hi.
Okay, so today we have in this room join our Vice Dean, Dr. Kuswanta Futas. Dr. Kuswanta.
Dr. Stephen.
Yes, and Dr. Spencer, nice to meet you. Good morning.
Morning.
Yeah.
He is our Vice Dean. So I'm so sorry that our Dean now in other meeting. So today our Vice Dean will join with us and deliver opening speech for today guest lecture.
Thank you very much.
Yeah, and also we are still waiting many students and also other faculty member join in this lecture. Please wait for a minute. Pak Sandi, gimana?
Ya, sudah. Dimulai saja, Pak.
Mulai aja, oke ya.
Nggak apa-apa.
Pak Warji, siap semua pasukannya?
131 partisipan.
Ya.
Ya, ini sebentar lagi.
Ini dalam rangka HUT-nya Pak Dek,
Ya.
mudah-mudahan lebih
Ya,
dari, hopefully kita lebih dari 300-400 ini mudah-mudahan.
Mudah-mudahan ya.
Mahasiswa kita, mahasiswa dari universitas lain, kemudian dari UGM, IPB, mudah-mudahan UB juga join. Dan, Pak. Hari ini padat juga Pak, di UGM juga siang ada international lecture, di mana-mana sekarang international lecture. Besok juga di tanah ya?
Di tanah, ya. Iya.
Ya.
Jadi Pak Dekan sama Pak Wadek harus siap-siap nih. Tiap hari ngasih opening speech ini.
Oh, ibadahnya besar itu nanti ibadahnya.
Oke, silakan Pak Waji, Pak siapa? Saya kira udah banyak ini. Yang lebih baik dimulai aja.
Sambil jalan aja Pak, terus saja. Ini banyak yang masuk juga kok udah.
Well, to all the participants, because it's been 9:40 a.m., we are about to start the international lecture. So for those of you who haven't turned on your camera, please turn on your and thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
First of all, I would like to say welcome to the third Diana Faculty of Agriculture, Lampung University, Dr. Insinyur Kuswanta Hidayat MP, the Honorable Head of Agricultural and Biosystem Engineering, Mr. Dr. Sandi Asmara, MSc, the Honorable Guest and Lecturer, Dr. Jani Stephen Jehia, and distinguished guests and participants. Ladies and gentlemen, welcome to the National Lectures on Agricultural and Biosystem Engineering with the theme Robotization and Informatization of Agriculture for Feeding 9 Billion People. It's a great honor for me. I'm Iqbal Santoso, will be your master ceremony for this extraordinary event. Ladies and gentlemen, before we begin, it will be nice for us to pray first, so that this event will run well from the beginning until the end. We invite all the attendees to join the prayer according to respective religion and beliefs. Prayer begins. Finished. Well, now we would like to introduce you to Head of Agricultural and Biosystem Engineering. So please welcome Mr. Dr. Sandi Asmara, MSc.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Ya pun.
Very good morning to all of you. Alhamdulillah, praise to God, the Almighty. Your Excellency Dr. Jani Stephen Jehia, invited speaker from Hokkaido Agriculture Research Center Division of Farming System Research, Smart Farming Group, Japan. Your Excellency Professor Dr. Insinyur Irwan Sukri Banua, MSc, Dean of Faculty of Agriculture, University of Lampung. Your Excellency Dr. Kuswanta Putas, MSc, Vice Dean of Cooperation Affairs, Faculty of Agriculture, University of Lampung. All faculty members, students, alumni, and ladies and gentlemen, the participant of the International Lecture on Agriculture and Biosystem Engineering, welcome to Lampung. Welcome to the Department of Agriculture and Biological Engineering, Faculty of Agriculture, University of Lampung. We are happy and very honored to host you all in this current international lecture. The main intention of the international lecture on agriculture and biosystem engineering held in our department is to provide an international academic atmosphere in our department and our university. This international academic atmosphere is very important to elevate our university reputation in global higher education. This lecture also will promote strong international research collaboration in our university. We believe that it will be a great contribution from our department to elevate our university accreditation. We plan to hold this international lecture ceremony. Today is the third international guest lecturer, and Dr. Jani is one of the key person in robotization and informatization of agriculture in Hokkaido Agriculture Research Center. Next month in December, our department has got an agreement with speaker from Pakistan. We also plan to invite many experts from Russia and other top universities to be our next guest lecturer. I hope not only our students, our faculty members, but also our alumni will enjoy this international lecture. Finally, our department would like to thank the Dean Faculty of Agriculture, the University of Lampung, for their continuous support for this event. On behalf of the department, I ask our Dean to officially open this international lecture. We wish you all have a fruitful and productive lecture today. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Next agenda is delivering speech that will be delivered by Dean of Faculty of Agriculture, Lampung University. That will be represented by the third Vice Dean of Faculty, to Dr. Insinyur Kuswanta Futasidayat, MP. Time is yours.
Thank you, MC. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
A very good morning to all. Alhamdulillah, praise to God the Almighty. Your Excellency, Dr. Zani Stephen Nehia, invited speaker from Nero Hokkaido Agricultural Research Center, Smart Farming Group, Japan. Your Excellency, Dr. Sandi Asmara, Head of Agriculture and Biological Engineering Department, Faculty of Agriculture, University of Lampung. All faculty members, students, ladies and gentlemen, the participants of the international lecturers on agricultural and biosystem engineering. Welcome to Lampung. Welcome to the Faculty of Agriculture, University of Lampung. We are happy and very honored to host you all in this current international lecture. I'm very happy to hear that the first and the second guest lecturers in the international lecturers on agricultural and biosystem engineering was held last September and October with successful lecture and fruitful discussion. Many researchers and students from various fields from Indonesia and abroad joined the lecture. In this third guest lecturer, Dr. Jani Stephen Jehia will share us about the latest technology on robotization and informatization of agriculture for feeding 9 billion people. As we know, it is expected that another 2 billion people is coming by 2050. Mostly, it will be in Asian and African countries. Indonesia, with a big population in Asia, should take a responsibility to provide a sufficient amount of high-quality food for feeding the next coming generation. For this reason, today's lecture is very important for us. I think all of us will enjoy today's lecture. Finally, I hope this international lecture will promote academic exchange and international cooperation between the two institutes of UNILA and Nero Hokkaido Agricultural Research Center Smart Farming Group, Japan, in the near future. The purpose of the cooperation is to establish long-term scientific and technical cooperative relationship, including mutually academic collaborative research and the exchange of information, techniques, and publication. Ladies and gentlemen, by the name of Allah, the God Almighty, I hereby officially open the third International Lectures on Agricultural and Biosystem Engineering. We wish you all have a fruitful and productive lecture today. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Well, thank you, Mr. Dr. Insinyur Kuswanta Futasihidayat MP, for delivering the speech. The next is delivering international lectures that will be led by our moderator. Our moderator was undergraduate student of Institut Pertanian Bogor, master student of Kochi University, Japan, and Doctor of Biosystem Engineering in Kyoto University, Japan. Also, he has plenty of achievements. He has published 27 journals, which the last one is Journal of Engineering Science and Technology, where the title is Use of Ultraviolet Spectroscopy and Chemometrics to Quantify the Percentages of Adulteration in Calcium Specialty Coffee. That's published on February 2021. And also, he has published 5 books. Well, please welcome Mr. Dr. Didin Suhandi, STP, MAGR.
Okay, thank you, Master of Ceremony, for the very nice introduction. Could you hear my voice? Okay, ladies and gentlemen, may I have your attention, please? So the third guest lecture is about to begin. I'd like to remind you all that during the lecture, the host will mute you all, and after 1 hour presentation, we will have question and answer session or discussion session. It's also about 1 hour. And so during that session, please ask your question by using chat box or raise your hand and deliver directly your question to the lecturer. Okay, Honorable Dean Faculty of Agriculture, University of Lampung, Professor Dr. Insinyur Irwan Sukri Banua, MS. Honorable Vice Dean of Faculty of Agriculture, University of Lampung, Professor Dr. Insinyur Purnomo, Dr. Amandamai, and Dr. Kuswanta Futas. Honorable Head of Department of Agriculture and Biological Engineering, Faculty of Agriculture, University of Lampung, Dr. Sandi Asmara. And of course, our Honorable Dr. Njane Stephen Njihia, our guest lecturer today from Nero Hokkaido Agricultural Research Center, Smart Farming Group, Japan. And distinguished ladies, guests, ladies and gentlemen, and of course, our beloved agriculture and biological engineering students. Good morning and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
First of all, I'd like to welcome you all to this guest lecture of International Lecture on Agriculture and Biosystem Engineering. Today's lecture is very special because it's also the celebration of our 25 years of agricultural engineering department in Lampung University. Okay, before the presentation begins, allow us to introduce our guest lecturer today. So Dr. Anjane Stephen Njehia, please, I'm sorry if I misspelling your name, Dr. Stephen. So Dr. Stephen is now a researcher at Nairo, Hokkaido Agricultural Research Center, Smart Farming Group, Japan. He was born in Nairobi, Kenya, and he completed his master's degree in 2016 and PhD program in 2019 from Kyoto University, Japan. He has much interest in development of precision agriculture for smart farming system using several advanced technologies, including spectroscopy, acoustic technique, and also ICT-based techniques such as drone and GIS imaging. So today we will enjoy the presentation. Maybe so many drone will coming in the presentation. He is now also one of young top researchers and has very active research in group on smart farming system. And it is a great pleasure for us to study with him about robotization and informatization of agriculture and aquaculture. Okay, without further ado, I'd like to welcome our guest lecturer today, Dr. Anjane Stephen Njehia, to deliver the presentation. The title of the lecture today is Robotization and Informatization of Agriculture for Feeding 9 Billion People. Dr. Stephen, screen is yours. Yeah, still.
Can you be able to hear me now?
Dr. Stephen, you are still mute. Okay.
Can you be able to hear me now?
Yeah, yes, I can. We can see. We can hear, but not yet. You share your screen.
Okay, just a moment. I will now share my screen, and can you be able to see my screen now?
Yes. Oh. Okay.
Yes.
Enlarge.
Okay. Thank you. Firstly, I would like to thank the Dean of the Faculty of Agriculture and also the Vice Dean for inviting me to give this lecture today. I also like to thank the head of department, and also I would like to also thank Dr. Didingsa for organizing this international lecture for today. Also, thank you, Dr. Unila. Thank you for today. Thank you for also Dr. Sandy Asmara and Didingsan. Thank you all for inviting me today. Actually, Dr. Didingsan, we were with him in the same lab at Kyoto University. So he is my senpai. Senpai in Japanese is a senior. So I feel nervous presenting in front of my senior, but
no.
I hope
you are one of my best kohai.
Yeah, so I hope to to not only share but also to learn more about what you are doing at the University of Lampung. So the title of my presentation today would be Robotization and Informatization of Agriculture for Feeding 9 Billion People. And as you heard from the introduction, currently, the population is increasing both in Asia and in Africa, and especially I come from Kenya. So this would be the outline of my presentation. Already, maybe my self-introduction has been done, but why I would stress this part is the population of Asia and Africa is increasing. So it is expected that in 2050, the population would be about more than 9 billion people. So that means our resources are being depleted, but we have to think about how to feed these 9 billion people. So coming from Kenya, I think similar somehow to Indonesia, where the population is increasing, we have to think of how to optimize our production. We have to think of not only robotization, but informatization of agriculture. So just a brief explanation is in my bachelor degree, I studied about water, environment, and soil and I was in the department of the faculty or in the faculty of agriculture engineering. So my basic is, soil or water and environmental engineering. Then from there, I went to study about sensing technologies so that I can be able to apply them for agriculture. And today, what I would like to share with you is just a few of what I have accumulated in this few years of research, as indicated from 2016 to about 2020. I would like to introduce especially about, firstly, NARO. Maybe some of you might not have an idea or already have an idea where we work at. So our research institute, also known as NOKEN or Noken Kiko, consists of, we do research and development from basic application for Japan's agriculture and food industry. And we also collaborate not only within Japan, but outside also. So we have maybe about 1,800 researchers, and currently also the number of foreign researchers is increasing. And previously, of course, we were in the Ministry of Agriculture, and now in 2001, we integrated to form 12 national and experimental organizations. So where are we located? So this is the map of Japan, and maybe already I think many of you might know, Tokyo is around somewhere here, center, and we are located here in the northern part of Hokkaido, and our experiment station is here, very small. That's the northern part of Hokkaido on the east side, which we are known as the Memuro Upland Farming Research Station. And I would like to share a few of or general information about the climate condition in Hokkaido, because this is related to the production of crops, especially in the northern part of Hokkaido. As you can see, our average annual temperature is about 6.8 degrees. I think much lower than maybe Lampung, your temperature, but also our mean annual precipitation is perhaps slightly higher. This is an average of about 888, and sometimes we have a maximum of 2,500 millimeters of rainfall. And our soils are mostly mixed between volcanic peat and heavy clay, but on the east side where we do experiment, we have mostly volcanic soil. What kind of crops do we produce here in Hokkaido? Generally, only in Hokkaido, Hokkaido contributes about 80% of total potato production in Japan. So that means only this from Hokkaido, we are able to supply 80% of all the potatoes that is used within Japan, and also sugar beet. Maybe in Indonesia you produce sugar from sugarcane, maybe, but here sugar is produced from sugar beet. Later we'll talk about it. So this is a slightly different, and it is only grown in Hokkaido, where supply is about 100%. And we also have a bit wheat and a bit of also half of dairy products. So why is this possible to produce this kind of crops in Hokkaido? Is because we have a large difference between daytime and nighttime temperature. So this ensures that crops receive a maximum amount of sunshine that is useful for converting for photosynthesis, in order to convert the crops products to either glucose or to starch. So this large difference contributes to maximum amount of sunshine for the crops. Another thing is we have a cool summer. I think maybe in Lampung you may have a humid summer or humid condition, but we have a cool summer with very many long days and long hours of sunlight. Also, mostly we have large scale production, large scale. That means size of the land is, most of the production is cultivated large, with a total area of about 1.147 million hectares. Now, I would like to divide my presentation to show examples of ICT-based farming techniques that are useful or perhaps might be of use, not only here, but also in Indonesia, through what we have, through the experiences or through the examples that we have. Now, we know especially about what is smart farming. smart farming will involve several items to make sure that we carry out farming activities that one will not be strenuous to the farmer and that will be labor saving. So first of all, smart farming involves activities that are labor saving. Why is this important? It is because labor for agriculture is decreasing, all over the world. Now, in Hokkaido, or in Japan, we have a problem with aging of farmers. So when we have this issue, it means we have to think about the issue of labor. Where can we get labor? So we need to produce activities that are labor saving. So to do this, there has been GPS automatic driving systems or use of multiple tractors in the field. Secondly, we have to maximize the yield of crops to ensure we not only produce high quality, but also optimize production. And then the activities we have to make sure that they are not dangerous or strenuous, especially carrying heavy items. Recently, there has been the use of assist suits. I don't know if you know about these kind of systems, assist suit, where if you wear this kind of assist, the amount of strength that you need to carry heavy items is reduced because of the mechanical condition of the suit that is able to assist you. We also have to make sure that even people who have no experience can be able to work in the field. Currently the use of most of the agricultural machines requires people to have a lot of experience, but by use of GPS or automatic driving systems, you can ensure that even people who have less experience can be able to carry out agriculture activities. Lastly, traceability. Traceability is a system where from production to consumer, the information about the product is clear. Recently, in Japan, sometimes when we buy rice, we can see the photo of the farmer. So people have a lot of trust that actually this rice came from this farmer. So that means there is traceability. Information can be traced back from the source to finally to the consumer. So what has enabled, especially smart farming? I would like to introduce about several smart farming guidance systems that are on the market. One, machine operation is dependent currently on individual proficiency. That means you need a lot of time or high amount of technique to be able to operate these machines. But currently, by use of GPS technology, such as shown here we have a Topcon or a Nikon Trimble or AgriInfo, AgriNavibus that is able to assist, especially with, for example, auto steering. So that means even without having much skill, the farmer can be able to have continuous movement of tractor on a straight field. Sometimes this is done by constructing what we call AB lines are shown here, straight line or curved line. So that means in curved line or slipper guidance, the farmer can be able to have continuous movement of the tractor. Also, currently, there has been wide working with boom sprayers or broadcaster. So why is this important? It is because the dissemination of the status of guidance system has now been well adopted in farmers. Currently in Hokkaido, we have many farmers using this smart or these guidance systems. Why? Because it helps them to be able to work out activities continually on a line. So that means when you are, for example, doing harrowing or when you are planting, you can ensure there's a straight line because you are using GPS guidance system. Now, for example, this one, Info Design, was released by one of our researchers who used to work in our group. And currently, it has a worldwide application of more than 400,000 people. And in Japan, about 20,000 people have been able to adopt this AgriInfo or AgriBus Navi system. Now, I would like to also share about setup of such a base station that enables, dissemination of RTK information. For example, this is a very simple system whereby using a U-blocks or what we call an F9P, that's a GNSS module, we can be able to set a base station where we can send GPS reference or correction information to either drones or agricultural tractors in the field. So to do this, you may be able to set up using what we call a network transport or N-trip system, where you can have a server or a caster or server where your information that has been collected from the satellite is sent to the server. And if you have a smartphone or a PC, you can be able to access the server and get correction information about your position. For example, if I am here in the office, I can be able to get RTK corrected information by connecting my maybe smartphone to the server, and I can get already corrected RTK information. So this kind of devices or this kind of base station can be able to be easily set up at a low cost price. Another thing that we would like to introduce that has enabled the utilization of a smart farming system is the implementation of what we call a mission planner. Why is this important? When we are working on large field, we also utilize the use of UAVs for agricultural activities. Now, these UAVs, you need to be able to determine the route or the pattern over which you are going to take images using the UAV. So to do this, information such as the altitude or the shutter speed or overlap ratio would be important to ensure precise, to ensure that you have enough overlap or enough stitching of images together. Because if you don't optimize this kind of altitude or overlap ratio, you might not be able to get uniform image of your field. And why are we using these UAVs? We are using them because we want to find information about the growth pattern of a crop. So that means information about not only tractor, but the UAV and how it will move would be important. So to do this, one of our institute here was able to develop what we call a mission planner. And you can actually see this online by putting in information about your drone and your altitude, the shutter interval of, let's say, one second, or overlap ratio of perhaps 70 or 80%. And then you can input information about your sensor. For example, you are using a camera with a certain focus length or F ratio of, let's say, 4, or angle of view and the shooting range. And then you can actually get the flight route or the grid pattern of your flight. And you can upload this information to your drone to ensure that there is correct flight path generation. Now, I would also like to share about another thing that has enabled the advancement of ICT for UAV is the use of viewer, this kind of viewer. So this enables close analysis of the field. As you can see on the left, these are orthomosaic images that were taken on the farm. And by using this kind of viewer, you can be able to see closeness. That's how close are some crops and the growth on each row or each pattern, and you can be able to estimate such a change over time. For example, when the crops are when the height is still low, until when the canopy cover is nearly about 70 or 80%. So this kind of time-based analysis would be important when you're working with UAV for taking images. Another thing that one of our researchers has also been able to develop is drone that is able to also take images. So that means currently most of the manufacturers, sometimes not only is the cost expensive, but also the camera, and you have to enroll to a service to take images. This is a drone that was developed by one of our researchers, and here this is our research center, as you can see. And bottom here is the image of the field where the multispectral camera is taking images. Such an aircraft would measure about this size and weigh about maybe 1.6 kilograms. This length includes the rotor also, and autonomous flight on autopilot with a flight time of about 20 minutes. And one of the issues we know with this kind of drones is the issue of batteries, because for a flight of approximately only 20 minutes, if you're flying for about maybe 100 meters above, you can cover an area of about 100 meter by 150 meter with quite a good spatial resolution of 2.4 centimeter per pixel. This will enable you to see information about the crop, such as the crop height or the length of the crop, or even you can be able to see the canopy cover change over time, as shown on this image. This is a time scale change of production of potato crops, and you can see with time that we can be able to monitor the coverage area. Look at this area, very low coverage, while some areas have higher coverage. And then during senescence, or what we call drying, you can see this area already drying very quickly. So this difference in vegetation cover can be able to be monitored. Also, by using multispectral camera, we can be able to estimate NDVR. That is the vegetative indices, such as shown on this image. You can see we can monitor the change in the vegetation indices of the crops. So not only are we able to do the 2D analysis, we can also be able to do 3D reconstruction. So when you have your image, not only do you want to get the canopy cover, you also want to get the information such as the volume of the crop. So you also want to get the automatic change in the height or other structures. For example, for potatoes, the growth of potato is under, but there are crops which we would have some of the maybe pimang or some other crops where we need to estimate, or even beans where we want to estimate the pods. So to do that, we might need to do a 3D reconstruction where we get the image not only in 2D, but we can get the 3D information of such a topography, and we can be able to detect height, or we can be able to record crop growth with time. Also, we not only can be able to get information about the 3D, but we can be able to get the canopy cover rate. For example, this is showing change in the growth canopy cover from from around June to the end of near harvesting. This is about September. So you can see that the numbers are from low 0.6 all the way to 0.9. So that's the canopy cover rate. We can be able to estimate how much cover was on sugar beet. And this is why this is important, is because this by estimating the canopy cover, we can be able to estimate the yield. Because if we have a higher canopy cover rate, then we have maximum absorption of sunlight for photosynthesis, and this translates to increased yield. So we want to estimate when is the canopy cover, what time is the canopy cover highest? So we can use this kind of tools to be able to determine the canopy cover. And after we have determined the canopy cover, our goal is to estimate the yield. The thing with potatoes is, as you can see here, the growth is under the soil, so you cannot be able to visualize from the outside. So what we need to do is use what we call the phenotypics, the information that we get from the sensor, such as the canopy cover or the volume, or we can even use the plant height. And from this information, we combine with the photosynthetically active information such as PAR. And by this, we can be able to estimate the dry matter production by photosynthesis. So we can use this kind of models to estimate and say, okay, this year we expect the yield will be about how many kilograms per meter squared. And to do this, we also need information about the environment, such as how much rain was there, how much sunshine was there during that period. So we can use environmental data into our model, and we can be able to estimate the dry matter of the potatoes. Now, another thing that has been very key, especially in estimation of potato production, we are not only concerned about the yield, but also the infection. Here, especially in Hokkaido, we not only have potatoes for yield, but we also have potato seeds. There are farmers that are specifically assigned to plant potato seeds. Now, what is the problem is in these potato seed farms? If there is a infection such as a virus, then if the virus has infested the field for about more than 4%, then all the potatoes are considered not usable. Why? Because they are seed potatoes, they would be transferred or they would be sent to other fields. So it means if there is some infestation of virus in the seed potato, and that potato is applied to another farmer, then there will be spread of that virus. So what is the problem? The problem is the detection of the virus is not easy. Normally, very experienced personnel have to go to the field and monitor visually and say, this area, there is infestation of potato Y virus, or we call it potato Y virus. And sometimes when you look at the leaves, you cannot tell, oh, this area has Y virus or this area has no Y virus, because it is not very easy to distinguish from our eyes. Therefore, one of our researchers is working on detecting this virus by using deep learning, where an image such as here shown on the left is put into convolution networks, where there will be first convolution or second convolution. And through these layers, at the end, we can be able to determine the properties of infected or healthy leaves. So some of the properties would include mosaic kind of pattern on the leaf. And using this for deep learning, they can be able to detect, oh, this leaf has virus and this leaf has no virus. So the goal of or the application of this is so that in the future, maybe the farmer might use wearable camera or take images by drone, and they can be able to ascertain, for example, this area is infected by Y virus. And what is the remedy? The remedy is all this area only potatoes in this area would be removed so that the infestation doesn't spread. What causes this virus? One is aphid, small insect called aphid. So if the aphid feeds on an area where it has already been infested by Y virus, it will feed on other parts of the leaves, and this will lead to rapid spread of this virus. Now, we have also been collaborating with other universities, not only here, but such as Wageningen universities. And here we are looking at estimation, or we are looking at the differences in varieties of potatoes, not only here in Japan, but those in Europe. So we grow them here and we determine the varietal or the differences in the variety. And to do this, we first of all have to determine their canopy structure, because potato itself or potato crops have a very complex canopy structure. I think all vegetation do. So determining the information such as the volume or the canopy area is important, because that will determine the health of the plant and will help us also estimate the yield. So to do this, we have been determining not only the volume, but also vegetation indices that are useful for monitoring the growth of this plant. And from this information, we can get chlorophyll content, or we can even determine the onset of disease, or how much is a cultivar tolerant to certain conditions such as harsh weather or even disease. So this is an example of, as I showed you in the previous slide, this is a time series change of a field that we obtained data. As you can see, it has actually mixed varieties inside this field. And some of the varieties, maybe you can see by the end, that's by near harvesting, still some remain green. So these have late maturity. So by using UAV, we can be able to determine the variety which has early senescence or that which has late maturity. And this will help in preparation for best time for harvesting, yield estimation, and also we can be able to know the cultivar ability to be able to withstand certain environmental conditions. We also take 3D models. We develop this kind of models for the field, where we hope in the future, even without going to the field, when we exchange information, we can learn about cultivar or a certain property of crop from this kind of models. And what kind of information can we get? For example, as you can see on my right graph, especially during maturity or during senescence, there is a very big difference in the cultivar in terms of maybe values such as NDVI. You can see this is in the period where emergence or the crops are emerging. And then vegetation is when, the leaves are increasing. And then during flowering is when we have flowering of the potato. After that, during maturity, there's a clear difference in the varieties. So we can be able to know, oh, this variety actually is ready for harvesting. This one is a little bit more, still, the growth is still increasing. This kind of information can be obtained from UAV analysis. And we can also get information about the harm volume or the total, the volume of the crop. As you can see here, some varieties have higher volume, some have slightly lower volume. And this is related to the yield. See, if you see this Euroviva, some variety like Euroviva has not only high volume, but also it translates to high yield, while some, which has slightly lower volume during the growth, it translates to lower yield. So this kind of correlation can be obtained from information that you obtained from the UAV by doing phenotyping. Now, let me go to another area where I want to talk about robotization. How much, now I talked about use of GNSS, and then we talked about the use of UAVs on the field. So tractors, working on the ground, and UAVs working in the air, because both kind of information is important. Now, I want to show an example of where we have, labor, the problem of increased labor or the need for labor being sorted out by use of AI-driven agriculture. So I would show some video here. So currently, this is a project that is being done for cabbages, harvesting of cabbages. And firstly, you can see that's a map of Hokkaido, and this is the location of the project. So what happens? This is general view of Hokkaido area, maybe for some of you who perhaps are yet to visit. This is just a general view of the field. And as you can see here, this is cabbages that are planted on such a plot. And what happens during this? This is a normal harvesting of cabbage. This is, we call it a cabbage harvester. And here, the implement here is cutting the cabbage, and then you have a conveyor belt, which is moving the cabbages up the conveyor, as you can see here. So one person is driving, and then the machine is cutting the cabbage, moves through the conveyor, and then you have about 3 people here at the back. What are they doing? They're removing the outer surface of the cabbage, the dry leaves on the top. Then they pack them, they put them in this kind of casing here, this kind of boxes here. Why? Because later these boxes would be taken, these boxes would be picked up. So about 3 people, and sometimes he has to stop and make sure that he is on the right line for harvesting of this cabbage. Then this kind of this kind of a tractor would come and be able to pick up these boxes of cabbages, as you can see here. So then pick them up. After picking them up, they are taken to a tractor, a trailer like this. Then they will be exported or taken to the nearest market. This is the normal routine. So you realize there are many people involved. One person has to drive the harvester and the cabbage harvesting machine. Another person has to remove the cover of the cabbages. Then another person has to come pick. So what if, we think about labor saving? One, driver, the person who was driving the harvester. So look at this closely. No one is here. No one is driving. So it's harvesting without anyone being there. So we have replaced him. So of course he can do another job. We don't mean that labor is lost. So what they can do, maybe if it is a farmer, they can be able to, there is so much work to be done. So maybe at that time the farmer can go at the back here, can go here at the back and help out with just removing the outer surface of the cabbages. So what is happening here is this device here is actually detecting the position of the cabbages and moving the cutter in the position of the cabbage. For example, I would skip a bit. So as you can see here, this is the detection. Each cabbage is being detected. Each cabbage. This is one cabbage, another cabbage. Then on top here, we have a line camera. So this line camera is able to determine the position for where to cut which cabbage. So firstly, the top camera determines which line of the cabbage to harvest from. Then the cabbage is cut. As you can see here, this is the detection. This is the cabbage which is to be harvested, which is here on green. So this bounding box is able to identify, oh, we need to continually cut this cabbage, and next would be this, and next would be this. So each cabbage is detected for harvesting. Then not only does it end there. So it is cut, as you can see here, and it is slightly taken up the conveyor. Each cabbage is taken up the conveyor belt. Then at the back, here you can see after the conveyor, we can determine, oh, one cabbage passed, maybe as RGB image, and also here you have a HSV, or you have a thresholded image on the right. Then not only that, another thing is, even the tractor coming to pick the boxes is automatic. So no one is driving it. It is just coming close so that it can be able to pick these boxes of cabbages. How? It is able to determine, oh, this is the docking. I need to dock here. This is like the docking station. So it comes close and comes to the docking station. After docking, what happens? No one is driving here. So it is using GN, that's RTK fixed information between this tractor and the receiver, and here the harvester. So it knows the actual position. Then automatically these boxes would be able to be moved on. As you can see here, they are moved on from the harvester to the receiver, so that it can be able to take it to the tractor, to the trailer, so the trailer can take the cabbages to the market. So we have reduced so many people. The machine is harvesting, then only 2 people necessary to remove the cabbage leaves. And here, no one needs to drive it. It can be able to come and dock, and then after it has docked, it can be able to move and now take the cabbages out. So this kind of labor-saving technique is important, or robotization for agriculture in our case. Now, we would also like to show not only cabbages, but also we have moving devices that are used to collect images. And sorry, some of the information is in perhaps Japanese, but currently we are not only taking information by drone or by not only harvester, we are also using this kind of close to the ground robotization devices. So here we have a RGB camera. You have this is called multispectral camera with different wavelengths, and here you have a depth sensor. Then we can be able to have such a device moving in the field as we collect data. Now this is a soybean. We collect information about soybean. So we can get a RGB camera giving us this kind of images, top image, side image, and we can also have multispectral camera giving us colors such as red, green, or near infrared. Then we can be able to estimate NDVI. Also, orange, cyan, or near infrared. So this kind of near the ground sensing, call it near the ground sensing. Why is it important? Because we can get much more detailed information that we cannot get from the drone or from the tractor. So from this near ground information, for example, this is the line we have soybean, and this is from the depth camera. And this kind of robotized small, in the future, I think this kind of small robots will be useful for the farm. Sorry, hours, I need to pull this. I hope to make it a robot in the future. Now I'm just trying to develop a prototype, but I hope in the future we can be able to have it as a, mechanized robot to collect information for us in the field. And what kind of data can we get? We can get like a 3D reconstructed image. As you can see here, this is 3D construction of such a plot. And this is 3D reconstructed multispectral information we can use it to get NDVI. This is also some 3D multispectral information about the So in the future, not only drone sensing, not only tractor sensing, but near ground devices will be useful for collecting information in the field for agriculture. Now I would like to move another another area that I worked on during my PhD or during my master. So we have now looked at large-scale farming. We have looked at, drone or in the air kind of sensing. But that is not all. We have to think of underwater sensing. Why is this important, especially for aquaculture? Maybe like also Indonesia, the effect of not only, let's say, religion that prompts some people cannot eat pork or some other food. And so aquaculture, many people, they actually eat fish and aquaculture products because they are not only healthy for us, but also because, their production of feed to protein for aquaculture is actually much more, higher compared to things like beef or other products. So, but what is most important for us is, for example, here in Japan, there's a lot of, marine fish production, also a bit of freshwater fish production. For marine fish production, we have about maybe 58% on yellowtail, the kind of fish, a bit on sea bream, and, salmon. And the problem is, as you can see on my right graph, feed is very expensive. Feed contributes to about 52% of all the production cost for aquaculture. Secondly, of course, is management, such as net or cage. It contributes to about 42%. And for freshwater fish, much more higher. Feed, 76%. Just like agriculture, also fertilizer contributes to, it's very costly. Now, what is the problem is when this kind of feed is used in excess, we have pollution. See this bottom graph. This is an image of taken actually in Japan where, around 10 cases of a year are reported. You see this white color here is dead fish from this cage. Why? Because of what we call a red tide, excessive eutrophication, or eutrophication causes red tide. So this causes many fish to die. So we are using excessive feed. Of course, we have to think about not only pollution, but feed is very costly. And in this feed, mostly it consists of carbon or nitrogen, about 16%. So this carbon leads to a lot of eutrophication in the sea. What can we do about this? Now, we are always told about, SDGs. We have to think about SDGs because we are being guided by these development goals. And by using these ones, we can, for example, the 14th one says we have to think about below water, life below water. We have to think about how clean we can ensure we carry out our aquacultural practices. Now, the problem is why there's excessive feeding, we don't know the size of the fish. For example, if you have a child and you don't know the size of it, you cannot just give them anything. Each food is based on their age, their size. So same for fish. Feed is just given in excess because farmers can only imagine, maybe the fish is this size. To measure the size of fish until today, manual recording, like the length, was taken. Another one is recently, this is a news that was in Japan. By using AI, image processing can help us identify the length or the size of fish. But I can imagine not all water is very clear for such image processing. Water is dirty or some sedimentation. So that means we cannot use only machine vision. So we have to think about new techniques such as sound. Why? Because it is not only dependent on, sound is not affected by sedimentation, but actually fish can resonate underwater when exposed to acoustic sound. So in this research, I try to develop what we call Helmholtz resonance technique. So by using Helmholtz resonance, we can be able to determine the volume of a sample. An example is when you have a PET bottle, maybe you have a drink and inside, when the contents or the volume of the drink changes, the pitch of the sound will change. If you, drink from such a PET PET bottle and the contents reduce, the pitch sound changes. Why that happens is because inside here, the content, the size or the volume, the air inside here oscillates because of the neck here forms a mass. And we know as a result of this oscillation, frequency occur. So that means if the content of the sample changes, then the oscillation, the speed of oscillation will change. The speed of oscillation of the mass on the neck will change. And what will happen? Frequency will shift. And using this technique, there has been already established techniques to estimate the density or the crack in watermelon, or maturity of watermelon. We can be able to detect also defects such as hollow defect in watermelon by using acoustic sound. And another researcher already applied such a technique to measure volume of sample in a conveyor belt. So we thought, if this technique is available in air, we can try to apply it underwater. And so to do this, we utilize a speaker as shown here, which is connected to a top cavity of a resonator as shown here. And then we have a bottom cavity of resonator here. So by using this top speaker, we generate resonant sound. And after this resonant sound is generated, it is sent to the cavity below here. And when the volume of the fish, or when the fish is inside such a cavity, then the resonance frequency changes. And one thing we also introduced was, as you can see, we do not have a microphone or receiver on this device. Why? Because the speaker is sending the signal, and also the speaker itself is able to receive the signal. Why? We use a special amplifier that is able to convert the signal, into the impedance. It uses an impedance technique. That means by sending the acoustic signal, we can also be able to capture the reflected acoustic pressure. And when we capture such a sound, then we can be able to have a higher signal, and less noise. That was one technique we tried to introduce. And let's look at it. This is a small fish. This is a very small fish, like we call them Japanese rice fish. Well, it's not the size of rice, but that is the name of the fish. And right is ugui, or Japanese space fish. And as you can see, as the volume of the fish is increasing, the resonance frequency is decreasing. And we can note such a decrease in frequency by a peak indicated here. You can see this peak is shifting. And when you do a second order derivative, you can see a very clear shift as the volume of the fish is increasing. Now, we can be able to also precisely estimate or precisely predict the volume of such fish by increasing the parameters of our device by looking at, let's say, losses that occur on the side of the cavity and accounting for these losses in the model. Then we can be able to have a precise prediction between the actual volume and the predicted volume of the fish. Now, as you realize, the resonator was very small. So, rice fish or 10-centimeter fish, it cannot be fully utilized by farmers. Farmers, we have like big tuna or big red sea bream fish. So how to make such a close to application device was another topic that we did. So to do that, we focused on measuring on the land, not only underwater, but on the land. That means if we have such a big tank, we connect a speaker on the side of the tank. By connecting speaker on the side of the tank, we can generate resonance cavity here. And when resonance occurs in this cavity, we can estimate the volume of total number of fish. This is the actual image of the resonator. This is the cavity, this is a speaker, and this is small tank, of course, about 5-liter tank. And inside such a tank, this time we have a microphone or hydrophone to detect sound. And we can capture resonance cavity and the volume of the fish. Now, what is important here is the frequency range is about 100 to 500. This range is okay for fish. Why? Because fish have a hearing range of close to 1000 hertz. So that means it does not affect it is not, acoustic noise for the fish because, beyond certain range of frequency, it can affect the lateral line where the fish detects changes in frequency. So we have to be careful of such a frequency range. And as you can see from our results, as the volume of fish is increasing, resonance frequency is decreasing, and amplitude also was decreasing. Now, we try to, commercialize, or it is now maybe the process for commercialization of such a resonator is underway. My previous university is still continuing on this research, and this is an example of Ehime, where we went to, aquaculture in such a cage, and we tried to estimate the size of, like in 1 ton, like a 1-ton tank, where we try to estimate the volume of yellowtail fish. That is a buri. We call it buri or yellowtail fish. And we could find, when no fish and when there is fish, there was some change in frequency. So actually, this can be used even in such a 1-ton kind of tank, or in the future, such a cage. The acoustic technique would be useful for measurement of fish in such a cage. And why is this important? To go back to my slide is because currently in Japan, feed is mixed with things like orange peels or, maybe olive oil or some olive. Why is this important? Because not only, we have we have a culture of eating, sashimi, or maybe Indonesia too, but people are also interested in, flavor in the fish. So feed mixed with some orange peel, when given to such a fish, then the taste would be somehow like orange, a bit flavor like orange. Why is this important? Because many people might say, oh, the smell of the fish is not so good. So by increasing such a flavor onto the feed, then it's given to the fish. So our objective is to know what is the size of the fish, so that we can determine best time for harvesting the fish with such a flavored fish. So this is important because this fish is very expensive. So this might be, add value to the farm, to the aquaculture farmers as well. And again, I'll finally go back to precision agriculture for post-harvest. So after we have, I started with drones in the air and tractors on the field, and we have worked on that. Then harvested products. After we harvest these products, we need to think about, post-harvest to avoid loss, post-harvest loss, transfer from the market, from the farm to the market sometimes take a long time, storage conditions. So we have to think about post-harvest techniques. So one activity or one research that I was involved with was mango. Maybe in Indonesia, mango is maybe very cheap. I'm not sure, but reading some maybe
the season now the peak season.
Mango Indonesia. That is very good. Here in Japan, we have such a mango, and this mango is sugar content is high, and you can see uniform red color. And some of these mangoes might be about 5,000 yen. I don't know how much that is in Indonesia.
Very cheap. Very cheap.
Very cheap. Right? So this is
now because the peak season, so now it's very cheap.
Yeah, because why they are costly is because the growth of each mango is regulated. Look at this image in a greenhouse. Each mango, farmer is monitoring, because why is this important? All of them are not harvested at the same time. Each mango is harvested different time based on its maturity. So what is important is to determine the best time for harvesting. Now, if you can see closely, these mangoes are covered in a white paper. So it's very difficult to know when is the best time for harvesting. Of course, RGB image might help us, but based on the, based on the farmer's experience, they can be able to determine such a best time for harvesting. But after harvesting, they have to be sorted, because each mango is based on a different grade. High grade mango, maybe higher price, some of them, higher price, slightly low. So sorting happens at a sorting facility. And what happens, maybe you can see here, these mangoes are being sorted, and there's a color chart. So look at the mango, the color of the mango based on the color chart, and then manually sort. This is very difficult because our eyes would get tired. We always look at red color. Maybe we would confuse somehow and mix them. So this kind of only looking at the eye and sorting is very difficult. So this is the color chart. How to differentiate A and B? They all look similar. B and C, maybe B might be on C. So this kind of color chart gradation might be very difficult to use. So one of the techniques we try to use is fluorescence technique. So by using UV light, we can be able to determine not only sort them based on color, but also determine some spot if there is some injury. For example, if you look at this mango from RGB image, maybe you think, mango is okay. But if you look carefully, there is some small dot here, very small spot. And with the time, such a spot become bigger and bigger and become bigger. So we have to be able to notice such small changes or such infection or such a black dot that is caused by insect or virus and be able to sort the mango correctly, maybe using RGB or fluorescence technique to help grading of the mangoes. And another thing that we thought of applying is, as I talked about previously, Helmholtz resonance technique might be useful for predicting the density or, because after harvesting, we want to know the Brix content, but it is very difficult. Some of, we have non-destructive techniques that are useful, such as NIR, that can be able to help us determine the Brix, but mango also has a seed at the center. So sometimes penetration of maybe NIR might be somehow not so efficient. So we might use such a sound technique because Brix value has a direct correlation with density. And by using this technique, we might estimate the volume of the such as kiwi fruit or mango fruit. By determining the volume and weight, we get density, and we can correlate density and Brix value. So this information might tell us what will be the Brix amount of such a product after ripening. So this is important. So we try to use it for mango, and we can find that, yes, there's some correlation between Brix content and the density of mango to prevent post-harvest analysis. So we can say, this mango would be best day to eat after harvesting would be maybe day 3 or day 1. We don't know. So we have to continually collect a lot of, we can say day 3 would be best for eating this mango because after day 5, correlation go slightly lower. So that's a information we can use for post-harvest analysis. But some countries, maybe like Kenya or maybe Indonesia, harvesting is one time, from the tree. So problem is there's a mixture between mature, immature mangoes. So after they are harvested and some are overripe because one-time harvesting. As you can see from this image, we have some are mature, some are immature. So we have to be able to sort and ensure that we separate between all these mangoes, some which are mature, some are immature. Then we can be able to get higher income for the farmer or higher and prevent post-harvest losses. So this is application of sorting of mango. And just to share some information, some of these techniques have already been commercialized by companies such as Ishida or Shibuya Seiki. These are companies that have also already worked on machine vision and they have commercialized, such as pinhole. This is a rotting of oranges or some pinhole can be detected by using fluorescence technique. So this is our post-harvest analysis. So finally, I want to show you some photos of Hokkaido, apart, where our research center is based. We have large fields. So here we have a very good season around July or June, where such plantations are planted with lavender flowers, different types of flower that form like a rainbow. Very beautiful. You can see huge fields with this graduation of color. And this is another area. Also, you can see like a mountain with very beautiful graduation in color. And these are landscapes where people have planted maybe various crops. And this farmer or this person decides to plant different colors of flowers that may give us, nice time to refresh away from research. We can take some time off and refresh from research. And this is a blue pond. And actually, it is not reflection. You see, the sky is very cloudy, but the pond is blue. you know, it's not reflection from the sky. It is because of some deposits of some, you know, aluminium or some compound coming from the rock under such a pond. So this is one of the attraction sites. You know, aluminium produces some colloid, and when reflected by sunlight, it gives us blue color. In winter, maybe Hokkaido, maybe many of you in, you know, Lampung, you know, it's very hot, but here in winter we have -20. You know, this is a same place. This is in summer. You know, this is in summer. You know, it is covered by water. Then in winter we can walk on the lake. That's amazing. You know, in summer we cannot walk because there is water, but in winter the lake is frozen on top. So you can see a child is playing and we can comfortably walk or try to learn some ski. And temperature is -20, but inside the house, sometimes 24 degrees. Why? Because houses have some double layer and people, you know, are always turning on the stoves to, you know, to prevent themselves from freezing. And maybe when Diding-san was here, we did some summer seminar together in the lab. I tried to find a photo, but I couldn't find the image in this photo. And yeah, I have also, you know, shared some of the results, and we have gotten some little award from our research. Thank you very much for listening to my presentation. Hi.
Okay, thank you very much, Dr. Stephen. Many information, it's amazing. So many information about precision agriculture you presented to us, from production to post harvest, upper soil, below soil, and underwater. It's amazing. So I think all of that suggests us that I think the precision agriculture and the utilization of robotization and implementation in agriculture is not an optional, but maybe an obligation for feeding the next generation, because as you say that another 2 billion coming people will coming soon, very soon, 20 and 50. So I'm and all of course all of us very curious to your presentation. So before I open and maybe for the audience or the participants to ask anything to Dr. Stephen, I have one question, Dr. Stephen, about your UAV drone. For so your drone is maybe a little bit different with what we learned in Kyoto, because in Kyoto is the area of cultivation is small and under sometimes is under the greenhouse. So the drone is small and the application is for harvesting small fruit, something like that. But your drone should be big, right? Because huge plantation. But how you change the battery of your drone? It is automatically change the battery, or because of course if we use drone, so we expect that the drone can operate 24 hours or maybe 12 hours, or maybe in the daytime. But for that we have to think about the battery, how to change the battery automatically, or of course big battery will be a problem because heavy, but small battery should be but it should be difficult for maybe changing the battery many times. So how about that?
Thank you for your question. We know now, you know, the limit is, you know, for UAV is the weight that it can carry and the battery time. Most of them now, some of them now have, you know, limit of about 20 minutes, but recently some have increased their time to 40 or 50 minutes flight time.
2 minutes. Oh.
Yes,
one flight.
Nearly 50, some of them have nearly 50 minutes. So today I presented what we call a quadcopter drone. We have what we call a fixed wing drone. Fixed wing drone. That's a, you know, it doesn't have propeller, because when you have a propeller, then the mechanical part means you need to have enough energy to rotate the motor for each rotor. But when you have a fixed wing drone, maybe I didn't share about it. We actually research and work with what we know with fixed wing drones. With fixed wing drones, you can be able to have a flight of an hour, nearly an hour of flight. So, and an hour of flight, you will be able to cover a lot of hectares. Maybe I think about 30 or 40. Fixed wing EBE drone, 100 hectares. See, we have a fixed wing drone that can be able to cover 100 hectares in an hour. So and why we are using and that one, you know, you can change the battery after an hour. And I think 100 hectares is quite a huge field to cover for taking images. So not only quadcopter, for large fields, you would have to use what we call a fixed wing drone. For greenhouse, you may even not necessarily need you may not necessarily even need a drone with battery. You may use what we call a swinging fixed wire. So if you have a fixed, you know, wire, then I think you may use it as either to charge or not necessarily need to change drone to battery, but have a fixed, you know, wires where you can take images of your plant inside the greenhouse. So you may not even need a drone inside the greenhouse. If you have fixed wires, you may take images on, you know, on a camera fixed on such a greenhouse. So that is the difference in the battery and the drone and the utilization either in greenhouse or large fields. I hope I was able to answer your question.
Okay. Thank you very much, Dr. Stephen. Okay. Please for other participants, you can using chat box to write your question, and I will read the question to Dr. Stephen. Or of course, you can directly ask to Dr. Stephen by. Oh, we have Dr. Ardian Ulfan. Please, Dr. Ardian Ulfan, one of our faculty member of engineering. He is also one of expert in robotics. Please, Dr. Ardian.
Thank you, Dr. Diding, and nice to meet you, Dr. Stephen. My name is Ardian Ulfan. I'm as told by Dr. Diding. I'm from the Faculty of Engineering, majoring in the electrical engineering. So, actually, in our department, can you hear me, Stephen?
Yes, I can hear you.
Oh, okay, thank you. Yeah. We produce some of the drone as well for the precision agriculture, both in the for the quadcopter and also the fixed wing. And as you said, we already make some mapping for the plantation, measuring in the sugarcane plantation, and as based on our experience, we can use the fixed wing, and we can map the plantation for 1 hour. We can got almost 200 hectares. But it also depend on the altitude of the vehicle.
Yes.
And also now we got the parameter when we fly in the altitude of 150 meters, so we can got the GSD of the image below 2 centimeters. Yeah, below 2 centimeters. And also it also depend on the quality of the sensor. Okay, but my question is not for the drone, because actually maybe we can discuss a little bit more, because we produce ourselves. The drone is produced by ourselves, from our labs. But I would like to ask about the biomass. Have you make some let's say the calculation of the biomass of a plant based on the image, because in some plants the fertilization time is depend on the weight of the plantation, on the plant. I mean on the plant. Yeah. For example, pineapple, when they try to make the fertilization time, they always use the weight of the plant. So, but right now, because we do not yet have some instrument to counting the weight of the plant, so they only use the age of the plant. So, okay. Do you have any experience about it?
So
For
thank
example,
you for your question. We
yeah.
We have not planted, yes, pineapple, but we have most of our products are, let's say, under the soil, like sugar beet or potato.
And potatoes, yeah.
Yes. Also,
Yeah.
we have been working on wheat, which estimating the wheat yield. And as you said, to estimate the yield, you estimate the biomass, and you can use. You're asking whether we can use the phenotyping property of the crop, such as later you use weight, but I can actually, for pineapple, I can maybe, if you're able to estimate the volume, of each pineapple. Why? Because from phenotyping, we can extract the 3D data. If you take your, I'm not sure if you take your, either by fixed wing or, after you get your images and you process them to orthomosaic, and later you can develop what we call a dense point cloud. And from this dense point cloud, you can be able to train or determine where is your specific pineapple. Maybe I can imagine pineapple grows in a crown and center.
Sorry. I'm not counting the weight of the pineapple, but I count the weight of the plant.
Yes.
Before
the plants, not the pineapple itself.
Yeah.
Not the pineapple itself.
Not the fruits. Yeah.
If it is not the fruit, then what we use, we use the canopy cover. We can use
canopy cover of the plants.
Yes, canopy cover of the plant, and there you can be able to estimate parameters such as leaf area index, LAI. And when you have LAI, it is a parameter of biomass, because from the model, maybe I presented some model before, if you have a LAI and you have PAR, you can be able to estimate how much is your biomass or how much is your expected yield.
Okay.
So instead of using weight of the plant, you can try canopy cover and LAI. It may give you some better, it may give you some better prediction for yield or for biomass.
Yeah, but our difficulty is when we use the canopy cover, it is quite difficult because it is not like the sugarcane. For the sugarcane, we can count, or we can notify the canopy. But for the pineapple plant, when it's getting higher, when they're getting high, they're getting older. So aerial imaging like the grassroots.
Like grassroots.
It's very very difficult to very
maybe
difficult
the density of the plants. Maybe Dr. Ulfan, the density of the plants is also influenced. Very dense. Yeah, I think because in pineapple production, the density is important. So they have to plant so many pineapple in same area. So maybe using group, how about the group of I mean in one area, so we select the group of plants, so that's the parameter to use to estimate the biomass production after harvest.
Yes, I think as Diding-san said,
or maybe after harvest, just after harvest, because
well, we need before harvest.
Before. Okay.
Yeah.
So I think what you
maybe
can
group of something area, so you can select just to estimate.
My suggestion is you can divide it into stages of growth. For example, you want to know, is it before vegetative start to increase? Is it the time you want to so you can, what I do with other crops, I divide into stages. Then I can know this is from this stage onwards, maybe I can use a certain variable, such as for example, in my case, I determine, from emergence, it is very difficult. From emergence, it's very difficult, for example, to estimate each individual volume because some leaves are very small. At that time, I may use a coverage. Then after a certain period, I develop what we call a densified point cloud. And from the densified point cloud, I estimate volume of, an area of, for example, plot. Then each plot. Then I can later use a vegetative indices such as NDVI or GNDVI, because vegetative indices after, let's say, maturity, they are very good for us because we can see difference in variety. So you can, why don't you try to divide into stages and then use each variable at different stage? It might maybe give us some more information.
Okay, Dr. Ardian, we have another question. So thank you for your contribution.
Thank you very much. Thank you very much. Yeah.
So hopefully you can communicate later deeply with Dr. Stephen, and we may collaborate agriculture engineering and Faculty of Engineering with Dr. Stephen to develop application in Indonesia. Okay, Dr. Stephen and Dr. Ardian, thank you. And we have 3 questions in chat box. First is from Dr. Maki. He's also a researcher, young researcher from Andalas University. You know Andalas University. Andalas is also same in our Sumatra Islands, but it is in the northern part of Sumatra. Okay. The question is it's also about drone. I know Dr. Maki is also researching in drone. The question is how to stabilize the platform or the drone during the flight to grab the image from the sensor without interference from the flight altitude. How to stabilize, because of course higher altitude it will be not stable, but something like that, because the wind and other factors. So how to stabilize the platform or the drone during the flight?
Thank you for your question. What we have now, we call it gimbal sensors. When you have a gimbal sensor, it is able to ensure that the camera stays vertically downward and without the influence of the movement of the drone. So I would recommend, currently, there are many gimbal assistants that are used to attach a camera. So what a gimbal does, it prevents horizontal or, this kind of, sideways movement and maintains your camera position. For example, in our case, when we have of course, it's not good to fly when the wind is strong, but when you have a slight movement of the drone, the position of the camera remains vertically downward because of the effect of the gimbal. So that is one way you can be able to maintain, to prevent the effect of the wind or the movement of the drone on the camera. So there are several companies that are now selling gimbals that are associated for cameras for drones. So I would recommend that as one of the solutions. Secondly, I think your flight plan is important. There are many, there are many ways. You may have, you may have what we call, the direction of, of your flying. For example, if you are choosing to have maybe if your drone, you want your drone to move in the same direction as your camera, or you actually, you want your camera to look sideways while your drone is moving forward. That will also affect, how much overlap or overlay you have for your images. So your flight plan is also important. I think best is fly at a height where, for example, you will cover your whole area, but still have very high overlap. So high overlap is key to prevent, let's say sometimes you have holes after you've made your orthomosaic image because you did not have enough overlap. So two things: good gimbal that will ensure the camera movement is not affected, and an effective flight plan of, let's say, front overlap of 80 and side overlap of, let's say, 80%, and making sure your shutter speed is also, at a good pace where you're able to take images of all your field. That is what I have in mind.
Thank you. Dr. Maki, any comments for the first answer? Okay, maybe we move to the second question, still from Dr. Maki. So if the drone fly in the medium altitude, such as 1,000 to 2,500 meters above sea level, so how to offset the Earth's curvature during image acquisition? Because yes, it's a little bit high, 1,000 to 2,500 meters above sea level.
Oh, I think maybe, I'm not sure, Indonesia, you can fly 1,000 meters. Maybe you are talking about height above sea level, not the flight of the drone. Here we have a limit of only 120 meters. We cannot go beyond there because of the effect of helicopter and security. But anyway, many softwares have a way to correct azimuth, or the effect of the Earth's curvature on when taking images. Yes, sometimes you may experience what we call a doming effect, where after you take your image, your land will be, like a slanting. So you have to do calibration. You have to do calibration of your camera. And also many drone processing softwares are able to correct for such effects. I think, I'm not sure, there are many commercial softwares that are actually out there that are good for analyzing the drones, and they are now specific on which sensor you use. For example, when you want to develop an orthomosaic, that's combine all your images to one picture, many softwares now have an option where you select your sensor. Why? It's because they want to correct information about that sensor. So that is one advantage. Secondly, I want to know if when they are taking images, are they also taking ground control points? If you have ground control points, then you're able to rectify, because you are able to do what we call re-optimization of your map by inputting information that you took from the ground. So we use GCP to be able to use that as reference for the ground, and we feed that information to our processing to ensure that we do not have ununiform ground surfaces from the orthomosaic. So those two things I would maybe recommend.
Okay, so I think maybe the drone itself, the development of the drone itself, and also the development of the program, or maybe image correction or something like that, will together improve the real application of the drone, I think.
Yes, and use of ground control points. Ground control points.
So first, of course, we waiting for the very advanced technology for drone, but also we are waiting for the advanced image processing for to correct the image, so it will be very nice, and then combine together, and then maybe more efficient and application for the drone. I think soon. So you are one of the researchers we are waiting. Please.
Thank you so much.
To do this. Okay, Dr. Stefan, thank you for the answer. And then we have another question still from Dr. Maki. So it's about the UV sensing. So you mentioned about UV sensing and also the fluorescence. So the question is it possible to apply it for outdoor application? So maybe in outdoor, we have so many interference from the environment. So how we fix if we apply this technology in the outdoor or maybe in situ application? So we bring our device to the sample, not bring our sample to our device to our lab.
Yes.
So how about this?
UV application relies on, we need to have UV light, because for UV, you are actually getting information about the product from the, excited light. So you need to, from the emitted light. So you need to excite your, light on your object. So because of the interference of sunlight, it becomes sometimes very difficult to, to apply use of UV on the drone. So one would be nighttime. So if we have a UV light at night, then we prevent effect of the sunshine. And if we excite such a light on crops or on your sample, then you may be able to get information. But another challenge would be you have to take images at a very close range, because for UV, excitation happens when the sample is closer to the, to the UV light. So that means if you have to fly your drone, you'd have to have it closer to the crop. But then you have effect of downwash from the rotor or from the propeller of such a drone. So we have, if it is within a greenhouse, then you may not necessarily need to use a drone. We can just use cameras on a wire or suspended cable. But outside, we would have to, rethink. If we want to get precise information, we have to worry about downwash. But the best time to take UV images would be at night for using application of UV on the crops. That would be maybe one idea.
Okay, thank you, Dr. Stephen. Yes, we have another question from in the chat box. But if any question from the live participants, you can raise your hand and directly ask to Dr. Stephen. Please, any question? If not, I will ask Dr. Stephen from the chat box. Okay, another question from chat box from Dr. Mona Batubara. He's also one faculty member from Faculty of Engineering. Dr. Stephen, I'm interested to the NDVI camera to use at a drone. Would you show or describe the specification? The specification of NDVI camera you use?
Thank you for your question. I will perhaps again, if possible, let me see if it's in my screen that I can be able to share information about specification of the camera that we are using. Just a moment, I check my slides. I found it. Maybe I will do, I'll share my screen now and see.
Yeah, please.
Okay, good. Just a moment so I can show. We have, what we are using is already a camera which is in the market. So not our very own that we develop. So this is already, maybe you can see my camera.
Yes.
You can see my slide. This is we utilize it's a drone that is a DJI. It's a Chinese manufacturer. It is called DJI Phantom 4 multispectral. It has 6 bands ranging from blue to NIR. And as you can see, when you have for NDVI, you need NIR and you need red to calculate your NDVI, as you asked in your question. So this kind of multispectral drone allows for those 2 wavelengths, which are necessary to calculate NDVI, which will be the subtraction divided by the addition of these 2 wavelengths. This is one example of multispectral camera that we use for imaging. For fixed wing drone, there are many sellers, like MikaSense is one of the sellers of the camera that you can check for their multispectral camera, which also has very good wavelengths for taking NDVI images for your drone. Of course, you can also there are many, I guess, customized multispectral cameras that will have wavelength based on the information you want to take. So if it's NDVI, if it has NIR and red, then you can be able to extract NDVI of your crops using such a multispectral camera. This was one of the camera that we used for the data that we showed for a multispectral camera. Thank you.
Okay, thank you, Dr. Stephen. how Dr. Mona, I think maybe later you can contact Dr. Stefan if you need to collaborate using the NDVI camera. But I think that camera is also maybe available in the market. Huh?
Yes, yes. Most of the cameras that we use are available in the market. We utilize some that are available. I think the difficulty would be combining each filter for each camera would be. But if you are able to do that, that will be much more, useful for Indonesia or for for research. That's much better because the information that is within each filter or each wavelength.
Okay, thank you, Dr. Mona. Any other question maybe?
Thank you very much, Dr. Stefan, Dr. Diding. Thank you very much for your information.
Okay, Dr. Mona. Hopefully, this international lecture also opened more collaboration research, more opportunity for us to work together. Okay, other question maybe from other participants. Okay, you can also, I didn't see any in the chat box, no question. Maybe from other participant, you can directly deliver your question to Dr. Stefan. Dr. Stefan is one of, very active young researcher. I know him because, he worked very well and conduct research. So, and also doing many collaboration with Kyoto University, Gifu University, and many university. And hopefully later with Indonesian University in Indonesia, Lampung University, of course. Any other question from maybe from Dr. Warji or our secretary? Any maybe comments or please, or maybe from Dr. Sandi. Okay, waiting for the next question. I have another question, Dr. Stefan. Yes, it's about the UV, the fluorescent technology. So, for fruit, for example, you mentioned the yes, we can separate the healthy and maybe unhealthy fruits from the beginning. In Lampung we have guava. Guava.
Yes,
guava is very popular in Lampung because we have huge plantation, not only pineapple but also guava. We call as a crystal guava. So one problem is the insect, the what we call as flying insect, something like that. That insect is come to the fruit when it's small, and then when the fruit, the guava fruit, become bigger and bigger, and inside, the insect is also grow up. And then when we open, actually the surface is smooth, the surface is okay, no problem, but when we open the fruit, then the insect is inside, so it will be very dangerous for our customer. So that's why we want to also separate by using image processing, UV or maybe UV camera or maybe NRI camera is maybe not easy to see inside. So is it possible to use your technology, fluorescent, to see the insect inside?
Thank you for letting me know about guava. As you said, the inside is difficult because most of the UV fluorescence techniques give us information about the outside. But I think one thing we can be able to study is correlation between inside and outside, because what happens is the pericarp or the surface cover of a fruit actually changes based on the condition of the inside. That's why when ripening normally starts at the center, and then if it starts at the center, then the color also changes. So if we are to determine, I would recommend not just one sensor, but a multi-sensor For example,
multi-sensor.
Yes, if we use fluorescence for determining the color of the skin, then we also compare between infected and not infected. If there is some correlation, then we can tell actually when there is an insect inside, it affects the surface color because we want to determine without cutting the fruit. And then second stage, we can determine, using NIR because, NIR has a very strong correlation, especially at maybe 710 or 20 for water, some peak appear. So we might use that to detect if, there is some effect of, this insect based on the, property of the NIR camera. Then I'm not sure if,
how about terahertz?
Terahertz would be
because if we open the fruit, the infected fruit has more water inside because infected by the insect. So is it possible? But because terahertz is very expensive.
It's very expensive. I think also with terahertz, the penetration depth is very, very thin. So terahertz, for example, can penetrate skin level or very, very thin penetration. So that means, I think, yes, thinking of terahertz would be good, but it's very costly and we cannot apply it online yet. So you may combine, if you say that maybe when infected, there's higher moisture content, then NIR is actually very good for, water, because we use it for determining the Brix content of fruits, because Brix content is also related to how much sugar and how much water is in the sample. So I would recommend one use of NIR sensor, which can be able to give us information. And if for terahertz, then for research, maybe close to application would take a bit of time because now currently terahertz only imaging, just realized now, only spectroscopy, mostly spectroscopy. So imaging of terahertz is a bit complicated. So I think we already have good NIR images. We can also use, of course, spectroscopy, and also we have fluorescence, both spectroscopy and image. So if we use these two, we can find some ratio of wavelength, maybe 800 and something divided by maybe 400 for UV. So this kind of ratio might give us information about insects. So multi-sensor and also multi-wavelength might give us much more better information.
Okay. It's very nice for the next collaboration. We have so many guava and you have so many sensor. So we combine.
Currently we are still developing. We just made a UV imaging sensor. So we are yet to build up more, so we can still collaborate in the future.
Thank you, Dr. Stefan. Okay, another question, please. We have still time. If you have any question about drone, about sensing, about precision agriculture, aquaculture, it's also very interesting because in Indonesia, especially in the rain season, sometimes we have problem with fishing, aquaculture fishing in pond because sometimes the water in moving from below to the upper part. And then the residue of the overfeeding come out from the below part of the water to the upper part. And then it resulted in many dead fish in a pond. So I think it's also important. Your technology may appropriate to feeding our aquaculture, especially in pond. Yeah, because many we have many ponds and also fresh water, freshwater fish. Okay, any question? Any question from other? Okay, if not, so maybe this is maybe enough for today's lecture. Yes, I think no other question. Also in chat box, I think because your presentation is very inspiring and very amazing for us, and many of us maybe now thinking about how to use that technology to real application, not only for research but also maybe for community service in Lampung and also on Lampung but in Indonesia in general. Okay, I think maybe that's all our to the lectures. Thank you, Dr. Stephen, and please give him a big applause, big hand for him. Thank you very much, Dr. Stephen, for your inspiring presentation. And maybe from later, our head of department will give you our appreciation. And of course, thank you for your coming to this international lecture. And hopefully we will arrange the next, oh yeah, already. This is the certificates of appreciation from us to Dr. Stephen, Dr. Anjane, Stephen, and Jehia. Thank you very much for your time. Thank you very much for your sharing today. And we hope more tight collaboration, not only research, but also maybe in education and in community service to us. Between us, between Lampung and Naro, especially with your group. Thank you, Dr. Stephen, and thank you for all participants for today's lectures. And I give the time and the screen to the MC, Master of Ceremony. Thank you very much.
Thank you very much.
Mr. Diding for leading the international lectures, and thank you so much for Dr. Anjane Stephen for the citation. And for the next, we have a session for to all the participants who may yes, we have a session.
Please turn on your camera. We will have a photo session with Dr. Stephen.
Well, for the first slide, 1, 2, 3.
Okay, move to the next.
Next slide. Next slide. Well, enough. Thank you so much. Well, those are our agendas for today that have been presented. Hopefully, it will be useful for us. And finally, representing for all the committee, I, Pak Santoso, would like to say thank you so much for your participation. And I'm signing out. Thank you and see you.
Thank you. Dr. Stephen, thank you. Thank you very much for your contribution.
Thank you very much for inviting me to join your international lecture.
So after Corona, I will invite you.
We look forward. I also visit.
Mr. Diding, just a moment, I want to ask for Dr. Stephen. How old are you?
Huh?
How are you, Dr. Stephen?
How
old are you? You look a baby face.
Yeah, still
nice
young.
to see you.
Nice to see you. Thank you very much.
Still young. Yeah.
Very energetic.
How old are you?
I am now 31 years old.
31.
31.
31.
Oh,
you have married, eh? You have married.
Yes.
Just I think last year married.
Yes, just last year. Yes, I just got
last year. So very young scientist, very active. So
yeah,
we hope
very smart. Very smart.
Very smart.
Okay. Thank you.
Yeah, welcome. I hope to discuss more with many.
Okay. Thank you, Dr. Stephen. Okay, thank you. Bye, Dr. Stephen. See you next time.
Thank you very much. Bye.
Okay, bye. Oke, terima kasih Bapak Ibu semuanya dalam rangka HUTEP ya. Insya Allah next month we will have the fourth lecture. nanti untuk remunnya bisa diremunkan di semester berikutnya.
Ini yang keberapa Pak Diding?
Ini yang ketiga Pak.
Oh ketiga, berarti saya baru 2 dengan ini ya. Berarti
Bapak Pak Soto ketinggalan 1 berarti.
Iya, yang mana dong?
Yang pertama kan dari UPM ya.
Ya ada.
Yang kedua dari Dalian University Cina.
Oh ikut semua kayaknya dong.
Nah ini yang ketiga dari Jepang. Nah insya Allah yang keempat kita sedang nego waktu dengan dari Jepang juga ya. Mungkin nanti Terahes ya atau nanti hyperspektral itu dengan BAU ya. BAU itu Bangladesh Agricultural University. Kalau waktunya cocok mungkin dua-duanya, kalau waktu yang cocok mungkin salah satu.
Tadi ngomong-ngomong si Jen berapa umurnya tadi Pak Ridwan?
31 Pak, 31 Pak.
Oh pantas muda.
Muda Pak, saya selesai
saya 62.
Ya.
Parahnya Pak Sabto.
Kan bisa dilihat dari wajahnya.
Ini gimana caranya makan-makan ya?
Pak Diding.
Ya.
Kalau ada yang polarimetrik Pak Diding.
Ya gimana Pak?
Request polarimetrik.
Oh gitu. Oh ya.